In this video, we are going to talk about micrococcal nuclease assays. So, micrococcal nuclease assays. And micrococcal nuclease, now what is that? So essentially, that is an enzyme that digests DNA. So unlike a restriction endonuclease, or at least the restriction endonucleases that we talked about in the last section of the class, which digested DNA with a specific sequence and only at a specific spot in the DNA, micrococcal nuclease will digest all the DNA. It doesn't care about the sequence. Now, a micrococcal nuclease assay, this is sort of a technique that is used to study chromatin, or I should say chromatin structure. Okay, so a little review on chromatin. So chromatin, now this is the complex of DNA and protein in eukaryotic cells. And the protein is traditionally histones, so histone proteins. And the most common group of histone proteins that are associated with DNA includes 4 histone proteins, H2, H2B, H3, and H4. Now we're going to have 2 of each of these to give us sort of this octamer of histones. So let's say this is H2A, H2B, H3, H4, and then we have one more of each of those behind here, and then we have this octamer of histones that looks something like that. And you'll see when we talk about centromeres that this H3 is sort of switched out for a different histone for the octamer of histones that are found at the centromeres, or a special type that is found at centromeres. But we don't need to worry about that for now. We just need to worry about the standard octamer. Now, around this octamer of histones is 146 base pairs of DNA. So that's how much of the double-stranded DNA, 146 base pairs, that will associate with the octamer of histones. And we will call this the nucleosome. Okay, so this is what chromatin essentially is made up of. Nucleosomes, DNA plus histones. Now, that's one nucleosome there. Now let me quickly diagram a couple nucleosomes here. So I'm going to put one there and one over here. And I'm going to wrap the 146 base pairs of DNA around there. And then I'm going to extend this, and I'm going to do the same thing over here. And then the DNA is going off the paper here. So this, the DNA between the 2 nucleosomes, is called the linker DNA. And you can see how this linker DNA is really long right here, relatively long compared to the nucleosomes. We're going to call this arrangement right here, we're going to call this open chromatin. So this is sort of an open structure. And sometimes scientists refer to this as euchromatin. Eu meaning true, so true chromatin. But we can call it open chromatin. Now, okay, so we have open chromatin, so you might be wondering, what about, is there a closed chromatin? So down here, I'm going to diagram what we would call a closed form of chromatin, which is also called heterochromatin. And you know what? I'm diagramming too much space between these. I didn't want to do that. Let me start over. Sorry, I don't mean to waste your time. Okay. Let's try again. Closed chromatin. So I'm putting the octamers of histones very close together. So the linker DNA is going to be really short, right? This is just one model of what closed chromatin might look like. And you'll see why this model supports data from micrococcal nuclease assays. Okay, so here we have closed chromatin, also called heterochromatin. So two terms here, euchromatin and heterochromatin, refer to open forms of chromatin and closed forms of chromatin. So, and you know, what does it matter? Well, you know, let's say if we had a gene in the genome, and that part of the genome, or where the gene is found, is sort of in this open chromatin state, you can imagine that it'd be probably, you know, much easier to transcribe a gene that is found in euchromatin than it would be to transcribe a gene that is found in closed chromatin. And so the mechanisms that cause chromatin to go back and forth between euchromatin and closed chromatin are not well understood. There are parts of the genome that are thought to always be sort of in this closed chromatin state, like centromeres and telomeres. And there's something else I wanted to say about that. And I'll probably touch on that later. Okay, so we're going to talk about how we can use micrococcal nuclease assays to determine, you know, whether a part of the genome is in the open chromatin form or the closed chromatin form. So, okay, let's say we have a question. We want to know what parts of the genome are in the open form or the closed form. So we are going to rely on this enzyme, micrococcal nuclease. I'm going to draw this enzyme right here like a little Pac-Man. And okay, let's say, so micrococcal nuclease, like I said, it digests all DNA, but there are some types of DNA it's not really good at digesting. In particular, DNA that is really close to the histones, in the octamer of histones, so in the nucleosomes. So I'm going to say no, it's not very good at digesting this DNA here. So here's the octamer of histones. And see this DNA around here, this 146 base pairs, it can't get to that. The DNA is too close to these other proteins. So micrococcal nuclease cannot digest that. So how about this right here? I have two octamers, and then I have just a little linker between them. And you know, it's not very good at digesting the DNA associated with the octamers, or that little piece, that little linker between there, because you know, it's kind of wedged in here. It can't get to that DNA. It just can't get to it. Let's say we're going to do this. It can get to it eventually, but it has a really hard time getting to the DNA between here. So we're going to say it digests the linker DNA slowly when there's not much linker DNA between these two nucleosomes. But it is really good at digesting this type of DNA. My octamers are getting sloppy. Sorry. So not good at digesting the DNA associated with the nucleosomes, but very good at getting the linker DNA when there's some space here, when there's a pretty good amount of space between there. Micrococcal nuclease can get in there and digest that. And the longer the linkers, the quicker it can get to that linker DNA and separate these two nucleosomes. So, okay, what can we do with that? Now, let's say we have a tube of chromatin, and how do you get chromatin? Now, we talk about isolating DNA a lot, and getting the chromatin is a little different. So what we have to do is carefully extract the nuclei from the cells, and then there are procedures to carefully take the chromatin out without separating the DNA from the proteins. So isolating the chromatin. Now, let's say we have chromatin. If we add the micrococcal nuclease enzyme to there, after about 5 minutes, we're going to have various fragments of chromatin in the solution, right? we're going to have some chromatin that looks like that is made up of 3, let's say 3 nucleosomes. We could have more too, so I'm just starting with 3. So let's say the micrococcal nuclease cut here and cut here. Okay, we have a unit of 3 here. So 3 nucleosomes with the associated linker DNA. And you know, maybe there'll even be lots of 2 subunits. So for the 2 subunit nucleosomes here, you know, the micrococcal nuclease got to the linker DNA on either sides of here, but it didn't have time, say, to get to this one right here. So that might be what we would expect after about 5 minutes. How about after 15 minutes? Well, maybe after 15 minutes, we'd expect, say, you know, maybe some of the some of the 2 units, you know, where micrococcal nuclease got here and here, but we probably expect a lot of these of the single units, just the single nucleosome units with the associated DNA that the micrococcal nuclease can't get to. And maybe, you know, maybe after 30 minutes or 45 minutes or an hour or whatever, maybe eventually micrococcal nuclease has enough time to kind of wedge itself in between all the nucleosomes and get to all that linker DNA. So all we're left with are the DNA associate. All we're left with is the DNA associated with single nucleosomes. So that we have millions of single nucleosomes in a tube of chromatin that has been completely digested into single nucleosomes. All the linker DNA is gone. Let's say this is like 30 minutes. Okay, so that's essentially how micrococcal nuclease works. But what can we do with that? So what we can do is we need to get the DNA after digesting it with micrococcal nuclease, and then we can check the sizes of that DNA by gel electrophoresis. So for instance, with this thing right here, what's the best way to explain this? Okay, so what we should do is, before we can check this, say the length of this by, and we could predict it'd be 146 base pairs, right? But before we can run that on a gel, what we need to do is separate the DNA from the histones. And then we check that DNA by gel electrophoresis. Okay, well, you're probably thinking, well, who cares, right? It's all going to be 146 base pairs. We don't need to do that. But, okay, there's something we can do. So just hold on one moment. So, now before we go on to a full experiment, let me just show you what gel electrophoresis results might look like. So, micrococcal nuclease plus chromatin, we're going to incubate for various levels of lengths of time. Then we're going to stop the reactions, and then we're going to get the DNA. We're going to remove those histones and just get the DNA. Now we're going to load the DNA on an agarose gel, and we're going to separate the DNA lengths by size. And let's say these are the lanes right here. And above the gel, I'm just putting these little tubes. Now these are our different micrococcal nuclease digestions of chromatin. Let's say this was the 5-minute time point. Let's say this was a 15-minute time point, and this was a 30-minute time point. So after 5 minutes, let's say we see 2 bands. We see a band right here. Let's say we say bands like this, 1, 2. So we separated the DNA, right? We separated the DNA by gel electrophoresis. So the smaller length DNAs are down here, the longer ones are up here, and we stained it with a stain, say ethidium bromide, so we can see all the DNA. And let's say we see a pattern like this. And this is a laddering pattern. And we see this little band right here probably corresponds to the 1 nucleosomal unit. And this DNA would be about 146 base pairs long. This one right here, this would be 2 units. This is 3 units. This is 4 units, and so on and so forth, you know, depending on how long we gave the micrococcal nuclease to digest that chromatin. After 15 minutes, we're going to see, oh, the 4 units and the 3 units, those are disappearing. So we're mostly down to 2 units and 1 unit. And after 30 minutes, maybe everything is down to 1 unit. And, you know, we could have our DNA ladder here, and we should see that the DNA here, this is probably 146 base pairs, and this is probably 146 times 2, and this is 146 times 3, and this is 146 times 4, plus the linkers that are left between the 2 units and the 3 units and the 4 units. But the most important part is we see this laddering, and the spacing correlates with the size of the nucleosomal unit. So what the Micrococcal Nuclease Assay is doing is allowing us to see, so the chromatin structure, right? It's allowing us to see the chromatin structure, and it's based, and the laddering pattern is based on the nucleosomes. Okay, so who cares, right? That doesn't really tell us anything. We can't really do an experiment just, you know, with this, you know, except to see that, oh yes, sure, there's a laddering pattern there, and the pattern is consistent with the length of DNA around the nucleosomes. Well, let's say we had a question here. So one point I should touch on just before we go on is that, and you might already realize this, because I think we touched on this earlier in the video here, is that micrococcal nuclease is going to digest this linker DNA very efficiently. But it's going to digest this DNA with the short linkers, and the nucleosomes are packed really close together very inefficiently. So we can use the fact that micrococcal nuclease digests this type of DNA in open chromatin efficiently, and this type inefficiently, to check how different parts of the genomes, whether they're in open or closed forms. So if you're confused by that, hold on one second. So here's an experiment. Let's say we are curious about in white blood cells. We have 2 genes. We have gene A, which is transcribed at high levels. Transcribed high, the up arrow is for high. And we have gene B, which is transcribed not at all. I was going to say low levels, but say it's not transcribed at all. And we don't know why this isn't transcribed. You know, we look at the DNA sequence and it has a nice promoter that we think should be active, and you know, we don't know why this gene isn't transcribed in white blood cells, and we're very interested in figuring out why. And one hypothesis is, well, maybe this part of the genome, for some reason, in white blood cells is closed. It's closed off in white blood cells. Maybe white blood cells, they don't need that. They don't need those genes in there, and it's they closed it off and put that part of the genome in heterochromatin. Well, we can test that hypothesis with the micrococcal nuclease assay. So what do? Well, we got the cells, then we get the nuclei, then we get the chromatin, and then we perform the micrococcal nuclease assay. So essentially we treat the chromatin with micrococcal nuclease in a time course, experiment where we're stopping the reaction after 5 minutes, 15 minutes, 30 minutes. Stopping the reaction, isolating the DNA, and then performing gel electrophoresis. So we take our micrococcal nuclease digested DNA and we split it. Let's say we're going to split it now. We're putting half of the sample, using half of the samples in one gel, and the other half in the other gel. And we're here are the lanes up top, so the DNA is going to migrate down this way. Smallest, shorter molecules migrating faster than longer molecules. And let's say here, we separate the DNA. This will be 5 minutes, 15 minutes, 30 minutes, and the other half of the samples will be over here. And when we run the DNA in both gels, we're not going to stain it with ethidium bromide because we don't want to see all of the DNA. We only want to see DNA that has sequence that is complementary, that is, we want to have the sequence for either gene A or gene B. So over here, what we're going to do is we are going to use a probe for gene A. This is essentially a southern blot, which we haven't gone over the details, but let's do one technique at a time. So on the right is a probe for gene A. So it's going to light up all the DNA sequences that match gene A. And over, I'm sorry, on the left would be gene A. On the right is a probe for gene B, and it's going to light up, cause to fluoresce all the DNA that matches gene B. Okay, so let's say over here, what we see is at 5 minutes, it looks something like this. Okay, we get this nice ladder pattern here. And then at 15 minutes, we see something like this. And at 30 minutes, we see something like that. Now, let's say for gene B over here, we see something like this at the different time points. Now, okay, so here's our data. So what's going on? How do we interpret this? Well, it looks like gene A is in a part of the genome that is associated with open chromatin, right? And micrococcal nuclease was able to get at all the linker DNA between the nucleosomes because the chromatin for gene A was open, was in an open conformation. So it was able to, over time, get to all that DNA and digest all the nucleosomes into single units. Get rid of all the linker DNA. But gene B, micrococcal nuclease, is having a really hard time at separating the nucleosomes. And the most likely explanation is that for gene B, the nucleosomes are really close together. So that would be consistent with our hypothesis that, okay, yeah, gene B is probably in a closed part of the genome. For some reason, that part of the genome is in white blood cells in a heterochromatic state, in a closed state. So that's how you that's how you could use a micrococcal nuclease assay to test certain parts of the genome to see if they are open or closed. And once you have that type of data, what do you do? What do you do next? So essentially, scientists are very interested in questions like that. What causes a part of the genome to be heterochromatic or euchromatic, and what are the proteins involved with keeping something open or closing part of the genome? What are the proteins, or I should say protein complexes, that open and close chromatin? And also, like, why would one part of the genome be closed? Why would it be heterochromatic in one type of cells and not in the other? So, what are the processes that control those things? So lots of other questions. I won't go into all those now. I'm sure they'll come up many, many times, various questions like these that we could test with the micrococcal nuclease assay. Types of questions we can test with this assay will probably come up throughout the course. I hope that video helped you understand the micrococcal nuclease assay, maybe more so than the notes, or maybe it complements my description of the process in the notes. So I think in the next video, I think we were talking about centromeres in the next video. So if you have any questions, make sure to come to the Q&A sessions, and if not, well, look forward to the next video. Okay, see you.